Off our next session by introducing our second next generation leader speaker for today, which is John Tuthill. Originally from Central Maine, John received his BA in biology and anthropology from Swarthmore College. He then had an interesting career as a cabinet maker and crab electrophysiologist before he went on to do his PhD at Genelia. Was the first PhD awarded from Genelia. I actually got to know him then when he was doing that work because I also worked in flies. He was looking at the algorithmic implementation of visual motion detection in the Drosophila brain. Very, very beautiful work. He then went on to Harvard where he did a postdoc with Rachel Wilson, and he pioneered the study of touch processing in sensorimotor neural circuits of the fly. John's now an assistant professor at UW at the Department of Physiology and Biophysics, and his lab combines genetic tools with electrophysiology and optical imaging to understand how the fly brain senses the body and controls behavior. So please welcome John Tuthill.
All right. Thank you, Saskia, for that introduction. And thanks for selecting me to be the kind of fly representative, the Drosophila person in the room amongst a group of otherwise mainly mammalian neuroscientists. I'm going to be talking today about the work that we've been doing in my lab. I started my lab last year at UW, just across the lake. And as Saskia mentioned, as a postdoc, I worked on touch processing in the fly. But since I started my own lab, we've actually gone in a completely different direction, which I did not expect. But now we're mainly studying how the sense of where the body is in space is encoded within the fruit fly nervous system. And I hope along the way that I'll point out a few cases in which things we have learned in the fly might be useful to the majority of people here who are studying the mouse nervous system or the mammalian nervous system more generally. Okay, so what is the sense of proprioception? So proprioception is often referred to as the sixth sense. It's the sense that we have of where our body is in space and how it's moving in space. So our bodies, our limbs in particular, which is what I'll be focusing on today, are innervated by populations of sensory neurons, like muscle spindles and Golgi tendon organs. And these sensory neurons detect mechanical forces in the body. They send signals into the spinal cord that signal the position and movement of joints. And so we have tens of thousands of these neurons distributed throughout our bodies. And collectively, they're kind of representing what it is that we're doing, what the motor system is executing as we move through the world. And an example of why this system is incredibly important is this patient, IW, Ian Waterman, who I think about 20 years ago had a really severe flu infection that essentially ablated all of the proprioceptive sensory neurons from the neck down. So he completely lost his sense of proprioception and touch in a sort of form of sensory neuropathy. And he was unable to walk or move essentially for quite a while until over the course of years he retrained himself to move his body entirely under visual control. So this is a movie of Ian Waterman, I think about 5 or 6 years after his infection, where he's learned to essentially guide his movements by looking at his feet. And if at any moment he were to look up or be distracted, then he would be unable to execute simple movements like walking. So cases like this, which are rare but do happen, demonstrate that this sense of proprioception that we often take for granted, because it is not often in the front of our consciousness, is really essential for how we move through the world and for the implementation of motor control in the nervous system. So all of the proprioceptors throughout our bodies project into the spinal cord, where they then connect to interneurons, and some of them ascend directly up to the brainstem. And then from the brainstem, they project to other parts of the brain, including the cerebral cortex. But understanding the sense of proprioception has been really tricky because recording from these circuits in the spinal cord and in the brainstem in a behaving animal is just technically extremely difficult. And in particular in mammals, we haven't had very good genetic tools for labeling the interneurons and the sensory neurons that are contributing to our sense of proprioception. And this is starting to improve as places like the Allen Institute build genetic tools to target particular cell types. But for the most part, we have a relatively poor understanding of how proprioception is encoded in the central nervous system, and then how it is used by the brain to control movements. So this is one of the reasons that we study this question in the fruit fly. But the reason I study the fruit fly is because I find them just to be amazing little creatures, and they're far more dedicated to things like crossing gaps than mice are. So this is work from Tillman Trefan, who was a graduate student with Roland Strauss, who worked on this kind of gap crossing behavior. And this is just a single example of how flies use motor control to achieve tasks that are relatively sophisticated compared to what we might expect from the size of their nervous system. So flies have a brain that's about the size of a sesame seed, or about the size of the volume of cortex that the EM group here is attempting to image. So there's probably about 150,000 neurons in the central brain. In addition to the brain that's located in the head, the part of the nervous system that we study is called the ventral nerve cord. This is the analog of the spinal cord in insects. And so in the ventral nerve cord, each one of these balls of neuropil corresponds to one of the 6 legs. So here, this is just an antibody stand against a synaptic marker that shows the structure of the neuropil. So in my lab, we use genetic tools to identify and label different cell types within the ventral nerve cord that we think are contributing to or processing proprioceptive stimuli. So like in the mouse, as has been described here, we have cell type specific tools that allow us to do things like express GFP or GCAMP or channel rhodopsin. One advantage of the fly is that we are able to do this much more quickly. So by contrast to the mouse, we have, at this point, around 20,000 different driver lines that label subsets of neurons within the fly brain. And instead of using a single promoter from a gene to identify a cell type, we often use intersectional strategies, like is now being done in the mouse. But we often will take multiple enhancers, for example, and intersect them in order to define a cell type. So when I talk about a cell type in the fly, like this cell type here, we know that if we go in and we record from each one of these cells, it will respond nearly identically to sensory stimuli. And then if we were to fill it and reconstruct its anatomy, they would be indistinguishable from each other. And if we look at the transcriptomic level, these neurons are also indistinguishable. So that's kind of the criteria we use to define cell types, much as is being done here. OK, so fly motor control is an amazing system to study, but it's also extremely difficult. And this exemplifies why. So this is work done by an electrical engineering master's student who was in my lab last year. And in this movie, he's using machine vision techniques to track a single joint of a fly as it's walking, and then just plotting the angle between the femur and tibia joints. So this being the femur, this being the tibia. So here he's just plotting the angle over time. So you can see that this is about a second. So the fly here is taking around 10 steps per second. They average between 10 and 20 steps per second when they're just walking along. And what we would like to do is understand how this behavior is controlled. So it requires that we measure things very quickly. So this tracking requires imaging with a couple cameras at around 1.5 kilohertz. But because it's so fast, that means that it's super interesting, because you need these feedback pathways to be operating continuously and very quickly. So that's one reason why we're interested in this system. And like in mammals, we know that proprioception is important for motor control. So this is work from Richard Mann's lab at Columbia, where they have a gait analysis tool where they are able to use a light sheet to detect when a fly's foot touches the ground. And then here they're just plotting the trajectory of the 3 legs across multiple steps. And so using some relatively imprecise genetic tools at the time, they found that if you silence a subset of proprioceptors in the leg, that the fly becomes much more uncoordinated. In fact, initially it won't really walk, but over time they slightly recover. And then you can see that their gait is much more jerky, and if you just look at them walking, they look unsteady on their feet. So we know that, like invertebrates, proprioception is really important for motor control of walking. OK, so what about the types of proprioceptor neurons that we find in the fly? So there are 3 basic types. I'm going to be focusing today mainly on these neurons, the cortisol organ, but I'll just briefly overview the 3 of them. So first, we have these things called hair plates. These are little sensory hairs that are embedded in the joints, or next to joints, kind of in the armpit of the fly. And so when the joint moves, it deflects these little hairs, and then they send signals to the central nervous system about the position of the joint. There are also these little dome-like structures in the cuticle that sense the load on the body. These are called campaniform sensilla. And then finally, the neurons we'll be talking about today are cortical neurons. These are basically stretch receptors that have mechanotransduction channels in their dendrites and are attached to the body by a tendon, so that when for example, a joint moves and it pulls on the tendon, the neurons are stretched, and they encode things like velocity and position. And these are in the case of the leg, which is what I'll be focusing on today, distributed across the entire leg. And just as an aside, I'm actually working on a review with Xeam, another one of the next generation leaders, actually a veteran next generation leader, so soon to be current generation leader, actually going to be leading. And we've been diving into the literature to try and understand kind of the analogies between proprioceptors in invertebrates and vertebrates. And so although the structures of the peripheral organs and the biomechanics of how they work are very different between invertebrates and vertebrates, what they're encoding is actually very similar. And so if I were to show a spike train from a campaniform cincilla versus a Golgi tendon organ, you'd have a really hard time telling them apart. And for the most part, these three classes of functional classes that encode velocity and position, load on the body, and then the position of joints are analogous between hair plates and Pacinian corpuscles, or joint receptors in this case, and Golgi tendon organs and campaniform cincilla, and then cortitonal organs and muscle spindles. So we think that the different functional types of proprioceptors that we find represent some sort of convergent solution to this problem of how you know where your body is. Like, these are the basic signals that you need to keep track of your body and to adapt your movements moving forward. Okay, so as I said, I'll be focusing today on these cortical neurons, and in particular on a subset of cortical neurons that are located in the femur and known as the femoral cortitonal organ. So this is 135 neurons. They're in the fly's femur, and they're connected to the tibia here by a tendon. So when the tibia moves relative to the femur, it stretches their dendrites. And then these neurons are, as I'll show you, selected for different parameters of joint movement, and they send their axons into the central nervous system. So this is a genetic driver line labeling the majority of the cortical neurons. So you can see their axons here coming in from the leg. And we have axons coming in from all 6 legs in this line. We'll be focusing just in particular on the axons coming in from the front leg. And then here we're labeling with a nuclear marker just the cell bodies in the femur. And so this is how we count the number of neurons we have there. And what you can see here, and I'll touch on this a little bit later, is that they're organized into kind of different compartments that we think actually have some functional meaning. Okay, and the way we're going to characterize these sensory neurons is using 2-photon calcium imaging. So we express GCAM6F in the cortical neurons using a GAL4 line that labels the majority of the cortical neurons. We also express TD tomatoes so that we can do motion correction on our image data. And then the way that we're going to look at proprioception is, first of all, to kind of take control of the animal's movements and to use this magnetic system that was designed by a postdoc at Kiramamiya, who's done all of the engineering here and the calcium imaging data I'm about to show you. So the fly is mounted in a little holder under the 2-photon. We glue a tiny little steel pin onto the leg, and we have a magnet mounted on a servo motor, one of the fastest servo motors that we could find or afford. And we basically whip the leg around from being fully extended to flex. And so we can track this. So all the traces I'll show you of leg angle are tracked from the video. We record this at around 1 kilohertz. And so you can see we have pretty good stimulus control. We can start with the leg extended, and we can move it to flexion and then back. And these kind of ramp and hold stimuli are what I'll be showing throughout the talk. Okay, so this is an example of what the data looks like if we look across this entire population of axons. So remember, we have 135 axons. Here we're looking at just a single plane. Go back and play that movie again. Okay, sorry, movie's not playing, but luckily we quantified it over here. So this is just the delta F over F of what's happening in this movie here. So what you see is that you start with the leg out fully extended, and then we slowly move it in this ramp and hold fashion to a flex position and then back again. When the leg is starting out extended, you see there's already some baseline fluorescence that drops away, and then it increases again when the leg is flexed, and then decreases again, and so on. And so there's clearly some kind of position modulated signal where the delta F over F is higher when the leg is flexed or extended, but on top of that, you get these faster phasic responses that are occurring every time the leg moves. So clearly there are mixed signals within this population. That's not totally surprising. This is 135 neurons. We don't expect them to be a homogeneous population. So we can use image segmentation techniques to try and pull out the different subtypes within the population that we find. And so this is a very simple way to do this. We just take all the pixels in the image, and we just compute the pairwise correlations between each pixel, and then we just do some simple clustering, like K-means clustering on top of that. And even with these very basic methods, you can pull out what appear to be functional groups that have some organization within the neuropil. So here, so this is what a projection of the two-photon stack looks like, and then these are the functional subgroups that we pull out from this clustering. So what you can see is that these green axons here, or these green pixels, I guess, are responding each time the leg moves. So we think of these as movement or velocity-sensitive neurons. And then there are a couple other populations, one of which appears to respond only when the leg is flexed, and the other which primarily responds when the leg is extended. And clearly, there's some mixing between these, because remember, we're imaging from a whole group, and a single pixel might actually have axons for more than one neuron. OK. And this is not the case in just a single fly. So here, this is group data across a bunch of flies. And this is showing that we see in each fly that we record in, we get neurons that are responding to flexion of the leg, extension of the leg, and then we have neurons that are responding to the movement of the leg, these faster phasic signals. And within the movement-sensitive ones, we see both neurons that are responding or pixels that are responding in both, when we move the leg in both directions or in a single direction. So we do see direction selectivity, and that's shown down here. So there are some that appear to be directionally tuned. Although we think that this is not a mixture of signals, and I'll discuss this later. We think this is real, that they're responding in both directions. OK, so at this point, we've done population imaging from a whole group of axons and tried to pull out the different subtypes that are there. But because we're working in the fly, we can use genetic tools to take this population and subdivide it down into smaller subclasses of neurons. And I use the word subclass here because, as I'll show you, these are groups of around, so this is around 30 cells. This is something like 20 cells. This is just a couple neurons. But we think there are further kind of subdividing cell types that you can find within these lines. But for the most part, what we find is that there are two basic groups or subclasses. One we call the club because of the shape of their axon terminals here. The other we call the claw because they form this very bizarre and mysterious claw structure. And then finally, I'm going to touch on a more specific line we have that labels what we call these fork neurons. OK, so first I'm going to describe the anatomy of each of these different subclasses. So we use this technique called multicolor flip-out. Those developed by Alyosha Nern at Janelia. This is work by Pralaksha Gurung, who's a technician in my lab. And she can stochastically label different neurons with different colors, and then she traces them. So if we take this very broad line we have that we used initially, we get neurons that look something like this. These kind of look like club neurons, and then we get neurons that look like claw neurons. From the claw population, these neurons look pretty similar to each other. Each one has this kind of tripartite axonal projection. And then the club neurons are a bit more diverse. You get things that appear to project intersegmentally, and then some that are just like a very basic club. And then these fork neurons are also a pretty homogeneous population. In fact, there's only 2 of them. So the reason we know the numbers here is that in addition to imaging their axonal projections into the central nervous system, we can go into the leg and actually count the number of cell bodies. So here, we're imaging from the cortical organ in the leg, and we're looking at the whole population. So this is labeled by a Gallifreyan that labels all cholinergic neurons. These are all cholinergic. And then we use the specific driver line to label club, claw, or fork neurons with a UAS red stinger, a red fluorophore. And then we can kind of combine these two and try and figure out how these things are organized in the leg. And for the most part, it looks like these claw neurons are distributed more distally, these club neurons are more proximal, and then this line actually only labels 2 cells that's kind of smack dab in the middle of this, this group here. OK, so the anatomy is interesting. Like they're different populations. They're anatomically different, but what we really care about is what they're encoding. So the reason we care about the club and the claw is that these are the 2 populations that are encoding movement and position of the leg. So these club lines, or these club neurons, respond each time the leg moves, and these claw neurons respond in a position-sensitive way, either to flexion of the leg or extension of the leg. So we think that these are the 2 groups that correspond to the position and movement sensitive groups that we saw when imaging from the whole population. So we've gone from kind of imaging a whole population, identifying different subtypes, functional subtypes, and then identifying genetic tools that label those particular subtypes. And this is just showing responses to different sorts of stimuli to kind of illustrate the dynamics of these neurons, the dynamics as filtered through calcium imaging, at least. So here, in these velocity-sensitive neurons, if you move the leg very quickly, they respond transiently in both directions, whereas these position-tuned neurons, when you have the leg extended, this group is on. When you flex it, they turn off. And these ones have the opposite response. So when the leg's extended, their baseline fluorescence is 0, increases when the leg is flexed. They stay on as long as it's flexed. This is 3 seconds, so this is quite a while. They're kind of tonically active. And then they turn off again when you move the leg back to its initial position. So in addition to looking at these subclasses, we found a few lines that label very, very small numbers of neurons. In particular, this line happens to label just 2 cells, and we can image from single axons. And the reason we're interested in doing this is that we want to know, as a population, we think that these neurons are encoding the kind of natural range of movements that the fly leg might be making. So how specific are the receptive fields of these sensory neurons? And what we find is that they're specific at least to the point where you get directional tuning in single neurons. So here, these neurons respond when the leg is moving from extension to flexion, and then don't respond at all in the other direction. And so we think if we were able to image from every neuron within this group of 135 cells, we'd see a kind of fractionation of direction, position, and velocity across the whole population. And that's a direction that we're headed in with this work. OK, so to summarize the sensory coding part of the talk. So if we look at the different subtypes of neurons that we find in this proprioceptive population, we see position-tuned cells. These are the claw neurons that respond to extension and flexion. And then we have movement-tuned neurons, some of which are bidirectionally tuned, some of which are directionally selective. So we think this is kind of the basic code for leg proprioception that the nervous system has access to. This is what it uses to control the body. And so the direction we're headed in next is trying to understand how the central nervous system takes advantage of these signals to do things like walk and cross gaps. Jim Truman, who is the kind of patriarch of Drosophila developmental biology, has shared with us a bunch of intersectional genetic driver lines that label what he suspected were second-order neurons in the ventral nerve cord. They're downstream of some of these sensory neurons we identified. So this is the whole population. There's around 12 of these in each segment, and then these are single neurons that were labeled using multicolor flip-out. So the structure of each one of these cells within a segment is pretty similar to each other. We think they're a relatively homogeneous population, and therefore we call them a cell type. So postdoc in my lab, Sweta Agrawal, has been doing whole cell patch clamp recordings from these neurons using a similar setup to what I showed you before. So we're using a magnet to control the position of the fly's leg. And in this case, instead of doing calcium imaging, she expresses GFP in the CNS and then uses visually targeted whole cell recordings to patch from these cells. These are hard experiments, and Sweta is a champ. So here's what Sweta has found. This data was sent to me yesterday evening, so it's relatively fresh, hot off the press. So this is a recording from one of these 14A neurons. Here, the leg is starting out flexed. When you extend it, you see that these cells depolarize. These are non-spiking neurons. There are interneurons that are very similar that are spiking. So there appear to be kind of analog and digital channels within this local circuitry, which is an interesting question that I'm not going to touch on too much, but I'm fascinated by. OK, and then if you move it in the other direction, so if you go from the leg being extended to flexed, then you get a hyperpolarization. And when you bring it back to being extended, it comes back to where it rested initially. So the interesting thing about these recordings to me is kind of how linear these responses are. So here, this response looks very similar to the stimulus that we're delivering here. So this pathway is more or less just reading out the angle between the femur and the tibia. I'm hard pressed I'd be interested to hear if people know of other situations where we have such a linear readout of a kind of external obvious parameter in stimulus space. So those were single traces, but sweaters record from a whole bunch of them now. And this population looks relatively homogeneous. They all are responding. They're all depolarizing to flexion, hyperpolarizing to extension. And then this is just a group of tuning curves showing their sensitivity across the full range. So for the most part, these neurons appear to start responding somewhere around 80 or 90 degrees and are sensitive primarily to extension. So this is only one of many interneuron subtypes that we know of in the Drosophila ventral nerve cord. So we imagine there's probably a group that's also encoding these lower angles, the flexion responses. And we also think that within this population, there might be some range fractionation even across these tuning curves. We just need more recordings to fully understand that. And then I just wanted to point out finally that we also have a movement-sensitive population of second-order neurons. So unlike the 14A neurons I described, these ones are responding transiently, much more like the club neurons I described earlier that are encoding movement. So to kind of put this all together, what we found in our initial foray into the circuitry of the fly ventral nerve cord is that we have separate channels at the sensory side and the proprioceptors for movement and position. And then we've been working on two populations, 9A and 14A neurons. These are defined by their developmental lineage because they were given to us by Jim Truman. But we think that there's probably something like 30 other cell types that are getting input from these proprioceptors. So we're only looking at a very small fraction of the circuitry at this point. And we have a little bit of evidence that these neurons some of them are connecting directly back onto leg motor neurons to mediate things like fast local reflexes, but they probably are also connecting to intersegmental neurons that are coordinating other legs, ascending neurons sending information to the brain. This circuitry appears to have a lot of parallel architecture for both fast local control, but also kind of long-range projections that are informing things maybe like navigation or course control. Okay, so to summarize, what I've shown you is that the sensory neurons encode position, velocity, and direction. The cortisol neuron population consists of several genetically identifiable subclasses, of which we are further subdividing them into subtypes, and that these different subtypes are tuned for specific joint parameters, and that they have distinct both peripheral and central anatomy that is characteristic of that particular type. And then we've started to work on some of the central circuitry in the second-order neurons that are encoding these proprioceptive signals from the leg. All right, that's my summary. Oops. So I also thought I'd just mention a couple other directions that we have recently embarked upon, which after the talks this morning, might seem sort of like bandwagons, but I think they're bandwagons for a reason, because I think that they're new directions that are going to be incredibly fruitful. So one of them is that we're part of a functional cognitomics project with people who have collaborated a lot here as well, Wei-Chung Lee at Harvard and Sebastian Sung at Princeton. And so our job is to do calcium imaging, much like the work that was being done in the Tullius lab. So we do, let's see if this movie plays, panoronal, or in this particular case, we're interested in motor neuron populations. So we image from all the motor neurons in a behaving fly. We then fix the tissue, and we ship it to our friends in Wei-Chung's lab at Harvard. They do serial section TEM, just like is being done here with the grid tape system. And then the data goes to Sebastian Sung's lab, and they're doing automated reconstruction. And then we do some proofreading and try and deal with the kind of existential dread of having that much data. And so this was I thought would be interesting for this audience. This is actually an image that Jasper, who is the grad student in Wei-Chung's lab who's doing the TEM. This is how we prepare the fly tissue for doing serial section TEM. So this is the fly brain here, and we actually embed it in a piece of mouse brain. So I don't know why they do it this way, and this is not a whole mouse brain section, but they found it a really convenient way to actually hold the tissue. So we use mouse brain to keep our fly brain, I don't know, stationary. And then we end up around the edges with a little bit of EM data of mouse brain. If anybody's interested in tracing in that, you can let Wei know. And then another project that we've recently embarked upon in collaboration with a couple labs at UW is to do high throughput RNA-seq of fly neurons. So we're using this technique seqwell, where, again, our role is to kind of pull out a bunch of neurons, dissociate large populations of neurons, and we do large-scale single-cell RNA-seq on that, and then we try and identify kind of different functional subtypes within that data. And in particular, the thing we're interested in doing is leveraging the cell-type-specific genetic tools we have in the fly to kind of recover identified neurons that we know a lot about as a ground truth for understanding both the cell types we can get out from these large data sets and to kind of quantify the variability both biologically and experimentally within these data sets. So these are both projects that are just getting off the ground, although as you can see here, Wei's lab has already imaged a fly ventral nerve cord. And we already have a little bit of this RNA-seq data, but these are things that will hopefully come online in a few years. Okay, so here is my lab. So Akira did all the calcium imaging in the cortical sensory neurons, and Sweta did all of the patch clamp electrophysiology. Pralaksha generated pretty much every anatomical image in my talk. And I want to thank our funding sources who are paying for this work. Also wanted to take this opportunity while I'm here to thank Kevin and Jack, who helped us. Kevin in particular came over and tuned up our 2-photon. He's kind of the optics guru of Seattle, as far as I can tell, and it's been really helpful. We've been recently doing 2-color imaging with his help, and so I look forward to interacting with other people at the Allen in a similar way. So thanks.
Great, thank you very much, John. If people have questions, we've got microphones, so raise your hand and we'll get a microphone to you.
Beautiful, John. What do you know or imagine about what synaptic transmission from that nice linear flexion sensor you showed the response of in the primary neuron? Is it?
We don't know much. We know that the linear one that I showed we think is GABAergic, and we have not yet done paired recordings between motor neurons and those interneurons, but that's something we'd love to do. So I guess a different way to phrase the question is like, does a motor neuron need a linear signal about where the leg is to drive its activity? From talking to kind of control theory friends, it doesn't seem like that's a great solution to controlling a leg. So I would hypothesize that there's some transformation that's happening at that synapse between the interneuron and a subsequent motor neuron.
Okay. Yeah, just quick question. Do you think that there would be different activity levels in your neurons if they were, actively moving opposed to passive movement?
Yes, definitely. And in fact, we've done a little bit of that. So the terminals of the sensor neurons get really heavy presynaptic inhibition from various other interneurons. And so in these experiments, the fly's awake, we're controlling the leg, but we don't control it perfectly. And so you will occasionally see the fly move its leg, or they'll try to move their leg. And when that happens, you see fluctuations that are a clear signature of some kind of presynaptic modulation. So we think that there's probably a lot of processing happening at the axon terminals of the sensory neurons as they come into the ventral nerve cord, which is really similar to like the work that Ayman has done in the spinal cord. Presynaptic inhibition in a system where you need really fast feedback control, one way to implement that is to do it right at the first synapse in the nervous system. So we think that whatever kind of modulation of that sensory input is happening as the animal is behaving, it's probably also predictive and interesting in the sense that they might be implementing efference copy right at the first synapse. Yeah.
Hey, John, were the movement-sensitive interneurons, I think they were 9A, were those spiking? Or those?
So there are two kind of subclasses, some of which are spiking, some of which are not spiking. Yeah.
So it would be overly simplifying to say the movement-sensitive are digital and position-sensitive are analog.
Yes, it would be overly simplifying to say that, but I wish we could. Sure, let's say that. Yeah, you heard it here first. Yeah.
Question up there.
So, hi, John. I was wondering if you had looked at different axes of motion besides flexion and extension. and how those axes are encoded?
We haven't, no. It's an interesting question. So the joint, it can move a bit around in a rotational axis. The stimulus control of how you would do that and at the same time that you're moving the joint is a little tricky. And so at this point, we've essentially been clamping it. But it's a really interesting question. So we don't know whether these particular sensory neurons, the way they're kind of mechanically set up, it's hard to imagine how they would detect rotation, but it's possible. Another thing to consider is that in addition to, like, there's a very slight rotation that the leg can do, and then there's flexion and extension, but then the animal also wants to know if its joint is moving in a way that it doesn't want it to. So there's this question that has existed for a long time in the invertebrate literature about how animals sense pain defined as something like that is damaging to the body. And so an interesting direction that we haven't gone in, but I'd love to, is to try and figure out which sensory neurons are detecting kind of aberrant movements of the body that are signaling when the body is doing something that it's not supposed to. So it's probably there, we just haven't looked at it yet.
All right, why don't we thank John again.